So now let's talk about culturing microorganisms. And what we're going to start off with is talking about a number of different types of um, cultures. And the first thing I want to talk about is the term inoculate. We've used this term a number of times, which basically means that we are going to have a sample of bacterium or microbes, I should say microbes, that are going to be placed or plated onto a medium and grown. Microorganisms that grow from such inoculum or an inoculate are called a culture. So, for example, this week we have inoculated 5 Petri dish plates to grow a culture of bacterium from our environment. Now, when we're growing these from environment, it's usually what we call a clinical specimen that we are getting from human material. So a lot of times whenever they grow a culture to check for strep, for example, they'll take a clinical specimen from your throat, and they will take and inoculate a Petri dish plate and grow a culture from such inoculum. Now what we're going to do is use a medium that we place it on. For example, the nutrient agar is the medium we used in our Petri dishes. There's a number of different medium, various broths that we may use, nutrient broth, and things of that nature. Now, when we're obtaining different types of specimens, clinical specimens typically aren't going to have just one bacterium in it, because we may have a number of different bacterium that we may have. When we're trying to go through, we try our best, go through and create a culture. We're trying our best to separate and isolate bacterium so we can then obtain what we call a pure culture. What is a pure culture? One bacterium. This is what Robert Koch was able to do, is to be able to isolate bacterium and to obtain a pure culture. If we aren't obtaining a pure culture, we may have a number of different types, and therefore it's not going to be as accurate. Now, when we are looking at our bacterium, our Petri dishes, here's our Petri dish, we're going to have all these different microbes here. Each one of these is referred to as a colony. We would also say that this would be CFUs, or colony forming units. The reason why we say this is because ideally this came from one bacterium to create the entire colony of bacterium that was grown. So we call these colony forming units. Now, what we performed last week was what we call the streak plate method. And it's one of the most common methods in which you can go through and create a pure culture, obtain a pure culture. This shows the steps of going through the streak plate method, which we did last week, where you obtain your sample and then you streak your plate in one corner. Then you get a sterile, new cotton swab. Ours were cotton swabs. Streak the second time. Again, new, sterile, streak, and then do it a fourth time. This way you'll notice how the bacterium gets spread out to where it will be isolated and individualized. You notice here they're very individualized. So that is streak plate. Pour plate method is another. And they're showing it here as so. Hopefully you can see that okay. You'll notice how they have gone through. And literally, you're going to take the agar. You put the bacterium in the agar while it's still a liquid and it's still fresh, and then pour it into your plated dish. And then you'll see the bacterium growing. And it'll grow in the middle, top, and things of that nature. It doesn't necessarily grow at the very top on the outside because we poured it in as we mixed it into the actual agar. So that's pour plate technique. It's a little different. Now, we also talk a little bit about complex and defined media. Honestly, I saved this for whenever we're going through the MSA lab and through the McConkey lab. We'll talk about defined and complex media, so I'm going to save that. We also have what you call slant tubes. So the Petri dishes that you looked at last week when we went through and did this street plate method is very common. However, we also use what's called slant tubes, which is basically a test tube that you see here, where we have gone through. I don't think you can see where I'm pointing at. Hold on. There's this slant tubes, and we have put agar in here at a slant in this test tube. This is on page 175, figure 6.11. And you'll see that we are going to use this, and you can put your bacterium on the actual slant. This is great because it shows different oxygen levels. And also you can put it down into what we call the butt of the tube. So you can stab the butt of the tube and go down in here. And that these are great whenever you're looking at oxygen concentrations of microbes and their growth. Again, I'm going to go through these other types of media when we go through and study for our MSA and McConkey lab. And then we'll start talking about the growth of microbial populations here in just a moment.